So today we're going to delve into the nuts and bolts of coronavirus vaccines because now, so this is early December 2020, and they're all over the news because first the UK and then Canada and hopefully soon the United States is going to approve the use of the first vaccine. And for the first time, we've known so far are press releases from these capitalist countries that are making these capitalist companies that are making these vaccines. And of course, a press release doesn't give you the raw data. And so you can't really see, all they do is give you their interpretation of their data. And so, but now yesterday in the New England Journal of Medicine, the Phizer Bioentec duo published the results of their first phase 3 results from their big trial that they started back in July. And so that came out yesterday. I read that paper, and we're going to talk about that, but we'll talk also about what kinds of vaccines there are in general and so forth, and then go on to some other things. This trial that they have instituted is pretty simple, really. And what they only wanted to know were two things. One, does it work? And two, is it safe? Now, you know, there are several different kinds of vaccines that have been generated over the last 200 years. As you recall, in one of my first coronavirus videos, I talked about the history of vaccination, beginning with Edward Jenner in England in 1798. And he was the first one to come out with the vaccine, and his vaccine was cowpox virus material, because they didn't know what viruses were back in 1798. And cowpox material, virus, basically was a living virus. And so that's the first type of vaccine. You can take a live virus, but it has to be attenuated, so it's not virulent, so it doesn't cause disease. And that's what he had with cowpox, because it was similar, but it was different enough so that it did not cause smallpox in humans, which would give you about 30% mortality if you got that infection. Now, other live vaccines are polio. That was the Sabin vaccine that came out in 1960. And also the childhood vaccines that we're all used to now, measles, mumps, rubella, those are all live virus vaccines. And those have all come from the virus, the virulent virus, that they've changed in the laboratory so that it's not virulent anymore. And there's a long process of that. That's a whole nother video that I have to make for you, I guess. So there's live, and of course, Pasteur thought that the only vaccines that would work had to be alive. And he came to that conclusion because he looked at the whole process as a microbiologist would look at it. And he knew that bacteria and microbes he could grow them in the laboratory, but if he put them on culture or gave them culture medium that would lack vital nutrients, trace nutrients, that the bugs wouldn't grow. So his major hypothesis as to how vaccines worked is that if you put the live bug into an animal or a person, it would deplete the animal or the person of that vital nutrients so that the bug could no longer grow. And that's what immunity was. And he was dead wrong about that. That's why I have a little problem with calling him the father of immunology. Anyway, what he was wrong about is that it doesn't have to be alive. It can be dead. And Jonas Salk knew that, because an example of a dead virus is the flu virus that we're still using today, that was first used back in 1942 when Jonas Salk was a young lieutenant in the Army, and he was in charge, or not in charge, but he participated in the immunizations of the recruits for World War II with the flu virus vaccine. And of course, at that time, we still didn't know what viruses were. And so they were worried that if they tried, they didn't have time to attenuate the flu virus. They could grow it up in eggs, fertilized eggs that had embryos growing inside of them. And so that they could grow that up in the eggs for a week to 10 days, crack open the eggs, get rid of all the big material and so forth, and take the supernatants from that. And then they could concentrate the virus and fix the virus with formaldehyde. So that killed it. And that was a dead virus. And that's And Jonas Salk knew that because the flu virus during World War II worked. And that's why Jonas Salk made the first polio virus that came out in 1955, 5 years before Sabin came out with the live virus. And there's a whole story about that I should probably take a whole nother video to talk about. But anyway, we've got the flu virus as a good example, and the polio virus with Salk as a dead virus. Now there's also what we call nowadays a subunit vaccine. And that means that it's not a whole bug. You take the part of the bug that you think is causing the disease, and you immunize with that part, so that it's not alive and it's not dead. They're just molecules, usually protein molecules. And an example of that virus or that vaccine is the DPT vaccine, diphtheria, pertussis, tetanus. And those are all three of those bugs are bacteria. But the bug itself doesn't cause the disease. It is a protein toxin that the bug releases that causes all the problems. And so back in the 1910s and 20s, they made a vaccine, the first subunit vaccine for those first 2 and then 3 of those bugs by just taking the supernatants from growing bugs and then purifying out the proteins from the supernatants, and then they chemically fixed them, or they heated them so that denatured the protein so that they wouldn't be toxic, and they called that a toxoid. So it was tetanus toxoid is what they used for their vaccine. So that's a subunit vaccine, and it works. It's good. You know, they say you have to get a tetanus booster every about 10 years. That tells you how good the vaccine is, and we don't get. And of course, tetanus causes lockjaw. We don't get lockjaw anymore, which is a very good thing. And then there's another kind of vaccine is a vector, what we call vector vaccines. These have been studied seriously since the onset of the AIDS epidemic in the 80s and in the 90s. And of course, I think, as I've said before, we still don't have a vaccine for HIV. But a lot of research has gone into this for the last 30, 40 years. And so by a lot of people, and a lot of advances have been made in the use of, in the generation of vaccines, and the vector vaccines. And the vectors, there's a whole myriad of different kinds of viruses and so forth, that what they do is they use genetic engineering, and they take the genes that code for the proteins or the gene product of interest, and they put it into the vector, take out some genes out of the vector, and they put in the genes of interest. And they also, at the same time, particularly in the AIDS deal, they worried. They couldn't say to themselves they were going to have to use these in individuals, and they didn't want to have the vector to be alive. So they crippled the vector. And one of the vectors that's had a lot of work on, and it's coming through the pipeline now for coronavirus vaccine, is adenovirus that's one of the viruses that causes the common cold. And what the advantage of that is that the vector has surface molecules that bind to receptors on human cells that gets, once you inject it into the recipient, the virus or the vaccine has to get from the fluid into the cell, and that's what the vector does. It acts as a carrier. It gets it into the cell, and then it uncoats and it releases its genes, some of which are coding now for the protein that you're trying to immunize against. And those are vector vaccines, and adenovirus is one of the major ones that they've used. But they've used vesicular stomatitis virus and all kinds of viruses that you probably never heard of. So those are all what we've had until now. And now we have a whole new category of vaccines that are called messenger RNA vaccines. And that's a new development. There have never been a vaccine that has been given to humans with mRNA yet, until now, essentially, because the two vaccines that are furthest along in the development of the coronavirus vaccines happen to be the messenger RNA vaccines. And there's a reason for that, and it has to do with being with the capacity to produce large quantities of the vaccine fast, and to generate the proper molecules, RNA molecules, and then do that very rapidly. And they could do that with the RNA vaccines, and it takes a little bit longer to do the old-fashioned vaccines. But the problem with RNA vaccines is because they haven't ever been used in humans, people are a little bit leery about them, because we don't know. We don't know the short-term, we don't know the long-term effects in humans. Now, they've been used in animals for quite some time now, and there's been, these vaccines have benefited from 40 years, 45 years of revolutionary science, molecular biology on the one hand, and immunology on the other hand, to be able to make these things work. Because there's several little subtle problems with using RNA as a vaccine. Number one, we have enzymes all over our body, on our skin and everything else, that are RNA aces, which means they chew up RNA if they look at it cross-eyed. And traditionally in the laboratory, I mean, you had to treat this like it was gold with gloves, and you couldn't, because you could inactivate your RNA as you were trying to purify it and use it in the laboratory. So they've been able to get around that, and they also had to figure out a way to get it inside the cells. And what they've done, they've wrapped it up in a fat coat, lipid. When they say lipid, nobody knows what that means, and so it sounds pretty good, but it's fat, and the fat then helps and they put the RNA inside that lipid coat, and the fat can attach to the cell membrane and sort of miscegenate itself right in the membrane of the cell, and then it can be invaginated and go inside the cell more easily. Now, they've also used different building block chemicals to make the RNA sequence, because the RNA copies the genetic code from DNA, and the sequence of the building blocks that are called nucleotides are what determine then the gene product, which is the protein which is going to be the vaccine essentially in the end. And so they've used synthetic nucleotides to build up their RNA molecules so that the RNA is translated into protein much more rapidly and much more stably than it would be if it was the normal regular old sequence. So all of those advantages, there was one more thing is that in immunology, they realized that the way the body reacts against infection by viruses is by the first line of defense, as we've gone through, is the innate immune response, which is primarily mediated by interferons that as soon as it senses viral RNA inside of a cell, it starts making interferon, and the interferon then goes out and protects all the surrounding cells because it shuts down protein synthesis, among other things, and really makes it difficult for the virus to replicate. Well, in the vaccine end of things, you want your messenger RNA to replicate inside the cell and to be translated and to persist for a long time, because you get more bang for the buck that way, because you put in one molecule of RNA and you may get out thousands of progeny, and then also bajillions of protein as resulting from that go out and circulate around in the body and stimulate an immune response, because then the immune system says, hello, that's not one of our guys. And so, it does all the good things that the immune system does to get rid of it. And so all of these things have gone into, and all of this very fundamental basic science has gone into making these RNA vaccines. And so the initial data are very promising, and it's been splashed all over the news that the Phizer, Bioentec, and incidentally, the Bio-N, it's bio and then capital N Tech, T-E-C-H, is a very small biotech company in Germany that had been working on the ins and outs of the RNA chemistry and the whole deal for a while. And it's a man and wife team that founded the company and started the whole thing and so forth. So if this all works out, they're going to become bajillionaires on the one hand, and hurrah for them. This is a good thing in the annals of science. The thing is that the paper that they've just now published that came out yesterday in the New England Journal is the first data that we've seen, that I've seen or anybody has seen, that shows you actually what's behind this 90% effective or efficacy claim and so forth. And this is really important now for all of us. You may not want to get involved in the weeds of all of this particular situation, but ultimately, I think the reason to spend some time on this first new vaccine is because there's a half a dozen or more vaccines. Well, I think there's like 40 vaccines that are in the soup or in the process right now. And so we got 7 billion people in the world, and we have to immunize everybody. So that's going to take a lot of vaccines. And so one vaccine is not going to be able to even cover the United States. And so so we may not, us common souls out here in the boondocks, we're basically going to have to take whatever there is after they inoculate the healthcare workers and the nursing home people and then all the people that are working in the private sector in really important jobs and so forth and so on. By the time they get to the common man, we may never see this RNA vaccine. But the particulars of the data from the trials are important, because ultimately we're going to have to be comparing one vaccine to the next, and the data from these trials, one vaccine to the next. Eventually, we're going to have to compare the data that are in the news and so forth with one vaccine versus another vaccine, because there's all of these different varieties of vaccines, types of vaccines that I just told you about are going to be used, and they probably are going to lead to different rates of efficacy, how effective they are, and also different toxicity or safety data. So the Pfizer Bioentec vaccine, they had 43,000 people entered into this trial that started the end of July, really the 1st of August. And the data that they've now just published is everything up until November the 15th. So it was a multi-center kind of a deal with multiple countries involved, multiple investigators, and so forth. So it was, to do that big of a trial, that's a huge effort. And now the reason they enrolled 43,000 people, half got the vaccine, half got placebo, is because there are two reasons. One is they would only need like 4,000 people in the trial if they were going to challenge the vaccinated and the placebo group, because that would be enough. You ought to give and then test for infection by testing for the virus in the nose and so forth. And that's what I talked about in my second video in terms of clinical trials to test for vaccines. And of course, that's unethical. We just must let the epidemic, roll on, killing thousands and thousands of people, 3,000 people today in the United States, because we have to consider our ethical principles. And of course, if you watch my second coronavirus vaccine, I think it's more ethical to have a trial where you have fewer subjects, volunteers, but that you challenge them. Anyway, we can't do that to make an ethical trial. Now we just have to release these people out into the environment and wait for them to become infected one way or the other. This Pfizer Bioentec vaccine, they gave them 30 micrograms in each dose, and they gave 2 doses 3 weeks apart, 21 days apart. The subjects in the trial, so one group there were 21,000 people, and another group there were 21,000 people. Let them go out into the environment, and they started. They then had the subjects report if they got any signs and symptoms consistent with coronavirus infection. And if they did, then they would then test those people for virus infection with the PCR test and so forth. So they called those, and if that came up positive, they called them confirmed positives. So if they looked at the data from 7 days after the second injection, they came up 7 days and then thereafter, and they followed them with a median duration of 2 months. A median is the number in the middle, which means you have the same number below that and the same number higher than that. And so after 7 days, they had 170 people infected out of 43,000. Now, if you do the math, divide 170 by 43,000, you find out that those 170 people are only 0.4% of the total number of people in the whole trial. So that's not very many. But the good news is that of those 170 that got infected, 162 were in the placebo group and only 8 were in the vaccinated group. So then if you divide 8 out of 170, you get 5%. And if you divide 162 by 170, you get 95%. So that's how they can come up with that number. And it sounds wonderful. And I admit, in other vaccine trials, that these are very early data. I think that's what I'm trying to say. And if as time goes on, and those people are out there circulating in the community and going to those bars and restaurants where they're likely to be exposed and so forth and so on, it's 170 is going to go up and up and up. And so I would be happier if there were 1,000 people, 1,700 people were infected within the same ratios. So the thing is, if the longer we go in this trial, we'll have more and more people that become positive. Now, if the ratios hold so that 95% of them are in the placebo group, that's great. But we'll see. We'll see what happens. Now, and so that's the efficacy standpoint of the vaccine. The other thing you want to know is, well, are there any what the doctors call adverse events? I love the euphemisms they use in medicine. Adverse event sounds like something good that you want to have. So anyway, an adverse event is toxicity, and there really only were 2 in this trial reported. Local reactions at the site of injection, and then systemic reactions or systemic toxicity signs and symptoms. The local will really boil down to pain, redness, and swelling at the injection site. And the placebo group really had none of that to speak of. I mean, 1 or 2, 3, 4%. It was only in the vaccinated group that they got, and they got 80% of the people in the vaccinated group said that they had pain on the injection site. Only 10% said that it became red, and only 5% said it was a little bit swollen. And the placebo group didn't have anything at all. And so right away, that this is not a double-blind study, because if you didn't get any local reactions, you probably were in the placebo group. But that's not. I mean, that's the kind of thing you get within the flu shot and whatever. And it just shows how sensitive your body is if you put something, and it also shows that the skin is a tremendous armor against the outside world. If something gets inside beyond the skin, and so that it does in this instance because of a hypodermic needle, the body reacts against that right away. And it's all due to these immunological innate and acquired immunity and cytokines and all the things that I've talked about. Now, the systemic symptoms really boil down to fever and fatigue, and there were some other ones that were very minor. But in fever, only about 5 or 10% of the vaccinated people got a mild fever afterwards, but 30 to 50% of the people said that they were really fatigued for a day or 2 after they got their shot. But none of those are life-threatening, and so that's all good. And this vaccine looks to be efficacious and also safe so far. Time will tell. We'll see what happens next. Now there's a couple of the other vaccines that are coming down the pike. One is Moderna, which is also an RNA vaccine, and is only about a week behind the Phizer Bioentec. And then after that, there's a couple of adenovirus vaccines that are really out there, and they're going to be coming through the news right away. So we'll see what happens. It'll be very, very interesting to see all the data from all of these trials. Now, there's one other paper that I wanted to just mention briefly, by a fellow by the name of Dan Baruch, who is at the Beth Israel Deaconess Hospital in Boston and at the Harvard Medical School. And this guy's a first-rate immunologist. He's one of the major players in the business. He got into it 15, 20 years ago with HIV and AIDS, and he's been into the vaccine end of things and the adenovirus end of things in terms of a vector to make a vaccine. And he's been working with Johnson & Johnson. And so that's one of the other vaccines that's coming down the pike soon. Well, at Beth Israel Deaconess, they had a primate center associated with them for years. And the major player in that whole thing was a young physician who ultimately, recently, within the last 5 years or so, got pancreatic cancer and died. And Dan Baruch has benefited greatly from that, because he's just come out with a paper last week in Nature using the primate center to do experiments with rhesus macaques. Now, rhesus macaques are the monkeys that are about, they're about whatever, 18 inches high. And they've been instrumental in many other development. I mean, initially in the Salk vaccine development phase back in the 50s. He's published a very nice paper showing the correlates of immune protection. That means that if we do, laboratory-based assays on the blood or anything else in the body of an immunized macaque or person, and we find a certain amount of immunological activity going on, is that predictive of being protected from the virus? And of course, in this instance with macaques, you can be sure because you can challenge the macaques with live virus after, so you can give them a vaccine and then challenge them. And you can look at the immune response first and then challenge them and see if everything matches up and so forth. And so he did basically 2 experiments that he reports in this Nature paper. One is he had macaques that had been infected with coronavirus and then recovered from coronavirus. He purified the antibody molecules from the serum from these macaques, and he transferred those antibody molecules in a dose-dependent fashion into other macaques, and then he challenged them. And so in this instant, he could show that the answer was yes, the antibody did protect against infection, but it depended how much antibody the animal had, the more the better. And so, but as you decrease the amount over time, then they were not protected. So that's very, very important data from the standpoint of using plasma from convalescent people to see if you can use it to treat people and or protect them from infection. So that's number one. So antibodies are good. Antibodies, I'm a T cell person, and so antibodies, whatever, So, but they're good, and how much you have is important, and what kind of antibodies you have is very important. The other thing he tested in his macaques was whether or not the T cells were good, and I like this part of the experiment probably better. And in this instance, what he did was he took macaques that had recovered from the infection, and he depleted the natural killer cells and as well as the killer T cells from these animals using antibodies. So you can take antibodies to these cells, inject them into the macaques, follow over time, and you can see the disappearance of these cells. And then he re-challenged these animals to see if they would, if the absence of those T cells would make them more susceptible to a reinfection, and the answer was yes. And so T cells are important, antibodies are important, and the amounts of these things are important in the animal model. And the nice thing, what it really shows is how scientists can use the experimental system whereby they can control the variables much better than trying to discern conclusions from human beings that are out there running around and going to bars and restaurants and so forth. So that's the end of this segment, and I hope you've gained from it, and we'll be back talking about more vaccines as time goes on. Thanks very much.
So if you've enjoyed this video, please like, subscribe, and sign up for my newsletter where I'm serializing my new book, which is called The Quest for New Knowledge. You'll find a sign-up link below. Hey, thanks again. It's been great.